Hello, my name is Haylie Kinman. I'm a fourth-year biological sciences major with a passion for animals. Today we'll be exploring the idea of does mimicry extend to flight behaviors in a mimetic damselfly? Prey species can evolve to produce aposematic coloration, which is a color signaling to let predators know about their unpalatability or unprofitability. A prime example of this would be the monarch butterfly, who's toxic for their avian predators to consume. Some prey species have evolved to copy these colorations in the form of mimicry, like the viceroy butterfly. Research done on this topic has been largely focused on the visual aspect, but the aim for my work is to expand our understanding of the evolution of multi-component aposematic signals to include behavioral mimicry, but more specifically, flight mimicry. To conduct this research, our study system was the neotropical mimicry complex known as the clearwing complex. The models in this complex are the Ithomiini glasswing butterflies, whose aposematic color signal is an ultraviolet reflective white wing patch. This patch is used as a warning signal to potential predators, which in this case are mostly birds. Some species of the neotropical damselfly family, known as Polythoridae, are known to display an imperfect mimicry of wing color, shape, and certain flight characteristics. We predicted that these damselfly species may also be exhibiting flight mimicry. We filmed the model glasswing butterflies and two species of the mimic damselflies, the Polythoridae and the Euthori fasciata. We also included two closely related species in non-mimic damselflies that served as outgroups and were without the white patches, the Cora damselflies that are related to the Polythoridae family, and the Heterina damselflies that are from a closely related family to the mimic damselflies. As a control, we also included glasswing butterflies without the aposematic coloration. We predicted that flight should be more similar between the model butterflies and the mimic damselflies than between the model and the non-mimic damselflies. To study flight mimicry, we filmed these insects in free flight within a field insectary. The insectary had 3 GoPro cameras installed on 2 adjacent sides and at the top of the enclosure. This allowed for the cameras to simultaneously record the flights from 3 different angles, giving us a 3-dimensional perspective that will be used when analyzing the data. These flight videos were then synchronized, calibrated, and manually tracked using Adobe Premiere and MATLAB. Flight videos used in this analysis were chosen based on the maximum amount of time the animal was visible on all 3 cameras. Once this tracking was finished, we then moved on to the analysis of the data collected from those tracked positions. Using R, we were able to analyze the tracked dataset as trajectories that would allow us to calculate a wide range of variables, including speed, acceleration, and sinuosity. A principal component analysis, or PCA, is a data analysis tool that takes a multivariate dataset and turns it into a smaller number of variables in order to observe the trends present in the data. A PCA allows us to explore all the variables collected and to utilize the variables that show the most variation. The first PC is mostly related to speed, which are the larger values of PC1, and sinuosity, which are the smaller values of PC1, while PC2 is mostly related to variation in the flight acceleration. The butterflies, shown here in blue, tended to occupy the areas of higher sinuosity and lower speeds, while the damselflies occupied areas associated with lower sinuosity and higher speeds. The mimic damselflies are shown here in green, while the control damselflies are shown in red. The data collected from the bidimensional plane show that the more sinuous flight of the butterflies only occurs when observed from the sides, but not from above. Moreover, the data also show that the male damselflies had a higher average velocity than their female counterparts, as well as a higher average acceleration from certain planes. Coming back to this PCA, a multivariate analysis of variance, or MANOVA, was ran on all of the PC scores, showing significant differences among the study groups. The Mahalanobis distances are a measure of the distance between the means of the distribution of individuals of each species, taking into account the variation among them. This tool is used to measure how many standard deviations away the study species are from the model. The distances are ranked from smallest deviation to highest deviation, where the smaller the distance, the more similarity between them. This figure is important for us to look at because it highlights how the study groups compare to each other. The mimic damselflies are color-coded in blue, and the outgroup damselflies are in orange, while the control butterfly species is in red. The ranking of the study groups by Mahalanobis distances suggests that the mimic damselflies tended to show more similar flight characteristics to the model butterflies than the non-mimic damselflies. However, there are 2 exceptions, the females of Heterina being closer to the model butterflies and the males of Euthori being far from the butterflies. Previous studies did not consider sexual differences in flight, but these distances show a higher similarity in the females in each species than their male counterparts. We predicted similar flight characteristics between the model Ithomiini glasswing butterflies and the mimic damselflies, and while there was large overlap between mimic and model species, there is also overlap between the model butterflies and the non-mimic damselflies. The data from the PCA analysis revealed that the damselflies typically had similar flight characteristics as other damselflies, and the butterfly group shared similar flight characteristics with each other. Our work also showed that each sex of the damselfly species shows quite different flight characteristics, with males generally being faster than females. Both male and female Polythoridae were closely located with respect to the model butterflies. Interestingly, Polythoridae damselflies are considered to be the poorer color mimics than the Euthori damselflies. Altogether, our results suggest that flight mimicry, if present, might be compensating for poor color aposematic signals in the damselflies. Our results might also be affected by our experimental setup, where the animals might be responding with escape flights when flying inside the insectarium. Future studies should focus on free-flying insects outside of insectary conditions, on detailed movements of the wings during each wing stroke, differences based on sex, and on how much emphasis predators put on appearance.